All right, awesome. Well, welcome to the video. This video is going to be all about the central dogma of biology, specifically molecular biology. So the main idea and thought process behind the central dogma is the importance of protein synthesis. So the goal of the central dogma is to make proteins. And this makes sense because proteins are so vital for human life and proliferation. We want our proteins to be made in an efficacious and consistent manner. So these following mechanisms are going to ensure that these proteins are manufactured in a correct and efficacious way. So replication is going to be started in the nucleus. So the main enzyme at play here is something called DNA polymerase. DNA polymerase is going to extend and elongate this sequence in a 5' to 3' manner. And other proteins and enzymes at play here might be SSBPs, helicase, topoisomerase, ligase, the list goes on. Next, we go to transcription. This is where we take the DNA and turn it into something called messenger RNA. So messenger RNA is more functional than DNA because it can leave the nucleus and head to the cytoplasm. From there, it can go to the ribosome where translation will occur. But we're still on transcription. The main enzyme at play in transcription is going to be something called RNA polymerase. This is going to allow a RNA template so that the 5' to 3' elongation for the mRNA can be made in an efficacious manner. So now that we have our mRNA, we need to make some modifications to it before it can leave the nucleus. And these will be in the form of post-transcriptional modifications, such as a 5' cap and a 3' poly A tail. These will prevent degradation in the cytoplasm. Finally, we have a process called splicing. Splicing is going to remove non-active and non-coding regions in the coding region called introns. So these introns are different than exons. So exons are going to be the main sites, the main active sites, and the main sites that are associated with protein synthesis. So keep the exons, remove the introns, the unnecessary introns. Cool. So at this point, we have our mature mRNA. We're heading out of the nucleus. We're in the cytoplasm, and we're heading towards the ribosome. So once we meet the ribosome, we're going to undergo a process called translation. This is where we're turning that mRNA into a protein, to a functional protein, right? So there's going to be 3 sites on the ribosomal complex, the A site, the P site, and the E site. So we're going to start with the A site, then we're going to transfer the amino acid to the P site. This is going to take energy. So what happens is ATP is going to be broken down into AMP, and that's going to catalyze this process. And from there, our amino acid chain is going to continue to be growing on the P site. So from there, our polypeptide chain is growing, and that is essentially our protein. And that should be it for now. I will make further videos detailing the intricacies of replication, transcription, and translation in the future. Thank you.